Welcome back everyone. So today we will learn how to do Western blot to detect a particular protein. So in this week we are going through this different blotting techniques. So there are mainly three types of blotting techniques: southern blotting, northern blotting and western blotting. So if you want to detect a specific DNA in a sample, then you have to go for southern blotting. If you have to detect a particular RNA, then you have to go for northern blotting, and in case of protein you have to do western blotting. So all the three blotting techniques have this basic theory. That means that you have to separate either the DNA, RNA or the protein by doing gel electrophoresis. And when you separate all the types of DNA, RNA and protein in gel electrophoresis, they are separated by the size. So then you have to transfer the DNA, RNA or the protein to a particular membrane. So that membrane basically absorbs the DNA, RNA or the protein, and then using that membrane you can detect your whatever DNA or RNA or the protein you want to detect. So these three kinds of blotting technique has the same protocol, and if you have go through the theory classes, then you will understand what is the main theory behind those blotting techniques. So in this lab we will go for this western blot where we will detect a particular protein. So western blotting is also known as immunoblotting because here we will detect the protein by doing antibody interaction. So there will be antibody interaction, and that is why it is also known as immunoblot. So in case of western blotting, there are few steps which we need to follow. So as I have mentioned, first you need to isolate you have to isolate the protein from a sample. So basically you have any particular sample, cell lysate or the tissue sample, and from that you have to isolate all the proteins that are present in case of that particular tissue or any other sample which you have. From that tissue sample you have to separate all the proteins based on their size, and for that we need to do SDS page, because as in earlier classes also we have mentioned, to isolate or separate the proteins in a sample you have to go for this denaturing SDS page, that is a gel electrophoresis, and the gel is made of polyacrylamide. So after you separate the proteins by doing SDS page, then the SDS page or the gel is placed onto a top of the nylon membrane or a nitrocellulose membrane or PVDF membrane. Now all this membrane basically take away the protein sample from the gel, and it retains the protein in the membrane. Now after you have blotted the protein in the membrane, then you can detect the protein in the membrane by doing incubation with primary antibody and secondary antibody. Since this involves this antibody, that is why it is also known as immunoblotting technique. So first step is to get the sample ready for doing the western blot. So in case of western blot, our sample will be proteins. So suppose we have cell lysate, you want to detect or want to see if a particular protein is present in a particular sample. So you have a sample where different types of protein will be present, and you want to specifically detect one particular protein. That is why western blotting is also known as western blotting is very selective, and it is also semi quantitative. You can detect a particular protein's presence as well as what is the amount of protein, the relative amount of protein that is present in the particular sample. So you have the cell lysate where different types of proteins are present. Then you go for a SDS page. Now for doing SDS page, the way we prepare the sample is similar to the normal SDS page. So basically you have to boil the sample with the gel loading dye, which will contain the SDS and bromophenol blue, and if you have cysteine, then beta mercaptoethanol or DTT. So that will ensure that the protein is denatured basically, and it is in the primary structure form. So the protein will separate in the gel based on their size. So not based on their native conformation, because we are running a denaturing SDS page. So after you prepare the sample, then you load the sample in the SDS page gel. Now in case of doing a western blot, you should always load a particular marker which is already pre-stained. So you should be able to see the marker. So marker means there is a protein ladder where different sized proteins will be there, and you can see those markers if it is pre-stained while the gel is running. If you do not use a pre-stained marker, then you cannot see where if properly the gel has been run. or what is the size in the gel. So till how you have to run the gel, it will be determined by the Preston marker. And while doing this blotting, when you are transferring your protein to a nitrocellulose membrane or PVDF membrane, you can also see that the transfer is okay or not, because you can see the ladder proteins because those are Preston, and you will be able to see those Preston ladder in the blotting membrane as well. Since the gel, we have to make a SDS page gel, and I have already done that, and I have already loaded the sample in the SDS page gel, and the gel is already running. So after you run the gel, another important thing for Western blotting is that you should not keep any lane which is empty. So if you do not have any sample, then just load loading buffer or the loading die, because if you keep any lane in the SDS page gel empty, then what will happen, that the lane side, at the side of the lane, if there is any protein, it will try to squeeze and try to, the gel will be very weird, and you do not want that. The size and the shape or the band should be proper. That is why you should not leave any empty lane in the SDS page. So it is a good habit, you should not leave any empty lane. So always load, if you do not have enough sample, always load some amount of loading die in the gel. Now after you run the gel, then we will go for this transfer. Now in case of transfer, you can go for this capillary action. So basically you put the filter paper, then the membrane, and then the gel, and all the filter papers are soaked in buffer, and by capillary action the gel, the proteins will be transferred from the gel to the membrane. But this will take a long time, and maybe from few days even. So to reduce the time, we will go for electrotransfer. So in case of electrotransfer, what will happen is, the way we separate the protein in SDS page, we put in a electric field where the protein will move from the. So during electrotransfer, what will happen is that the protein will come out from the gel, and it will go to the membrane and get stuck to the membrane. Since the membrane is towards the positive side, the negatively charged protein will go and stick to the membrane, and then we will take out the membrane, and then we will go for blocking. So it is important to always block the membrane. So what is the meaning of blocking? So basically here what we will do is, so the proteins that are present in the gel, it will go and get stuck to the membrane, but there are a lot of space in the membrane which will be empty, because there is not everywhere in the gel the protein will be present. So there are empty sites in the membrane, and if you do not block and go for directly for this primary antibody incubation, then what will happen, the primary antibody should able to bind to the protein of your interest, so it should be specific. But if there is any kind of empty space, since it is a membrane and it can it has affinity towards protein, then the primary antibody will also get attached to these empty spaces, and there will be a huge background noise. So the primary antibody will detect your protein, as well as there will be other places where you can see the antibody, because it was empty in the membrane and it will get stuck to the membrane. So to reduce the noise in the membrane or the gel, you have to go for blocking. So in case of blocking, we go for this non-specific protein is incubated with the membrane. So what will happen in any place where the membrane does not have your protein or any protein, then that will get the non-specific protein will get absorbed to that particular membrane, and then when you go for primary antibody incubation, the antibody will specifically bind to the protein of your interest, and other places, since there is already blocked by different types of protein, it will not get attached to it. And when you detect the primary antibody with the help of secondary antibody, you will only able to see your particular protein of interest and not any other places. So blocking is an important step in case of Western blotting. Then you have to go for different kinds of washing. So you have to wash the gel and go for this primary antibody incubation. During primary antibody incubation, the primary antibody is raised against your protein. So the primary antibody will selectively detect your protein, and it will get attached to the primary your protein of interest. But you cannot detect the primary antibody, that is why we need the secondary antibody. So after the incubation with the primary antibody and several wash steps, then we will incubate the membrane with with the secondary antibody. Now secondary antibody is raised against the primary antibody. So the secondary antibody will bind to the primary antibody. So in this way your protein will bind to say primary antibody, and the primary antibody will get bind to the secondary antibody. And in case of secondary antibody, there is always a detecting molecule that is present in case of secondary antibody, either that is a HRP or horse radish peroxidase enzyme or alkaline phosphatase enzyme. So different types of enzymes are there attached to the secondary antibody. So after binding to the secondary antibody, you give a particular substrate for the particular enzyme that is present in your secondary antibody, and depending on the substrate you can detect the position in where your protein should be there. So wherever you see the detection band, that is where your specific protein is there in the membrane. And depending on the amount, if you are using a peroxidase enzyme or a phosphatase enzyme, there will be a light production, so chemiluminescence will be there. So depending on the amount of light that is produced by the secondary antibody, or basically the enzyme that is attached to the secondary antibody, you can also semi-quantitatively measure how much protein you have in a sample. So basically if you have different types of sample, you run different samples in the same gel, and in the same gel you try to detect the amount of protein that is present, and quantitatively you can see how much protein is there in a different samples, and also it will give the qualitative measurement and the size of the particular protein, and if at all the protein is present in the sample or not. So Western blotting technique is, that's why used in different kinds of detecting tool. So in case of HIV, if you want to get the particular protein present in a patient sample, blood sample or not, you can detect that particular protein using Western blotting. There are different disease patient where Western blotting helps to detect any particular diseased protein that is present or not in the sample. So thereby Western blotting is a useful technique for selectively detecting and semi-quantitatively measuring the amount of protein is present in the sample. So to begin with, here I have already started SDS page gel. So here I have loaded my sample, it is a normal 15% SDS page. I have loaded my sample and along with a pre-stent marker. So now I can see the marker and how long it has gone, where it is there, and I can, depending on the pre-stent marker, I can stop when I want to stop the gel. So before I stop that, as I mentioned, there is a membrane to blot. So mainly two types of membranes are used, nitrocellulose and PVDF. In case of nitrocellulose, it has very high affinity towards the protein, but the problem with nitrocellulose membrane is it is very fragile. So you cannot, it's basically it will break soon, or you cannot handle it properly. So PVDF membrane is a great choice for this kind of method because it is very sturdy and it also have higher affinity. Another thing is for nitrocellulose membrane, the noise, the background noise is very less, but in case of PVDF membrane, if you are not careful enough and if you skip the washes or not properly do the washing steps, then there will be huge noise. So in case of PVDF membrane, you need to do the washing steps thoroughly. So reduce the background noise while detecting your protein. The another difference between nitrocellulose membrane and PVDF membrane is that in case of nitrocellulose membrane, you do not need to activate the membrane. It is already activated. Whenever you want to do the blotting, you just soak the membrane in the transfer buffer and place it on the top of the gel. But in case of PVDF membrane, you need to activate the membrane using methanol. So you have to activate the membrane for about 1 to 5 minutes in methanol, and then you put the membrane in a transfer buffer. So transfer buffer usually contains trace to maintain the pH, glycine, and a small amount of SDS it might contain. And also 20% methanol it has. So the methanol will help to retain the shape of the gel, so do not skip the methanol part in the transfer buffer. So mainly the transfer buffer has trace, glycine and methanol. So since I have this membrane which I am using, this is a PVDF membrane, so I need to activate the membrane in methanol. But So there are two types of membrane you can buy in case of PVDF membrane. Either it's a pre-activated membrane or you need to activate in the lab. So this membrane which we got is a already activated membrane, so I don't need to do further activation. But you should always check the label when you are buying the membrane. If it does not say that it is activated, then you should go for methanol treatment. So you have in a small box you have a methanol, then you put the membrane in methanol for some time, and then you take out the membrane and put it in the transfer buffer. So before we go for this blotting, we need to equilibrate the membrane as well as the gel in the transfer buffer for some time. So while my gel is running, I will put this membrane, since it is already activated, I don't need to put it in the methanol. So I will put this membrane in transfer buffer. So I have in this small box, I have small amount of transfer buffer here. It is stress glycine and methanol. Another important part of this technique is that you should not touch the membrane or the gel by bare hands. If you do that, then there will be some noise and you will get blotchy gel. So using a forceps, I am taking out the membrane and I am putting this membrane in the transfer buffer. So if your membrane need to activate in the methanol, so if you do not activate the membrane in methanol and directly put in the transfer buffer, you will see that the membrane is not getting soaked in the transfer buffer. So that time you will know that you need to activate the membrane before putting it in the transfer buffer, because it is a hydrophobic membrane, it will not get soaked in the transfer buffer. So here my membrane is already pre-activated and it is already soaking in the transfer buffer. So I will keep the membrane in transfer buffer in a rocking condition for some time, and in the meanwhile we will take out the gel and also put the gel in the transfer buffer. So I have put the membrane in transfer buffer to equilibrate the membrane for 10 to 15 minutes. In the meanwhile, we will take out the gel from this plate. In a small another box, I have again to transfer buffer, and as maybe you have noticed that I have a very small size I have cut. So in case of this membrane, they come as a roll. Also in a pre-cut version we have it as a roll, and depending on how much you need, so basically there will be 2-3 lanes I will need only. So depending on the measurement of the gel where my protein will be there, I will cut a small piece of membrane from the roll. Because membrane is very expensive, do not waste the membrane, do not cut the full membrane as a whole. Now we will take out the gel from the plate. So I am stopping the SDS page run. As you can see, the marker has been separated properly, and I know when my protein will be there. So I have to stop now, and according to the marker, I will also cut the gel into small fragment, because not everywhere the protein is present. So this is a plate which we got. Here the protein is present here. Now I will separate the plate. So this is the short plate we have, and we'll keep it here, and the gel is stuck to the bigger one. So now I will just cut my gel according to the size I want. So first I will remove this top or the stacking gel basically. So you don't need the whole gel, don't go for this whole gel blotting, because then you will need a long or much more membrane, and as I mentioned, the membranes are quite costly. I don't need the whole gel, so I will just cut the gel. Using this cutter. So this we will discard. I will keep the ladder because that will ensure when the transfer is done, it will tell me when, if the proteins are transferred properly or not. Lower portion also I will cut. I can also check the size. So these are the things which we got from the membrane. So my membrane is of almost this size, so the gel should be a little bit smaller than the membrane. So now that I have separated out my gel, I have cut it into the smaller fragment. I will put this gel in the transfer buffer to equilibrate. So these are SDS page, so you don't need to be very careful, don't break the gel and put it in the transfer buffer. And we will keep it in the rocking motion, the gel and the membrane in the transfer buffer for 10 to 15 minutes, so that our membrane and the gel is equilibrated in the transfer buffer. Okay. So 15 minutes is over. So now our membrane and the gel is equilibrated enough in the transfer buffer. We also got this tissue paper basically. So these are the blotting papers, and this is not normal general tissue paper. So this is a high quality blotting paper or Whatman blotting paper, and we will soak this blotting paper as well in the transfer buffer. So after this we will go for this transfer. So we will make a sandwich. So just keep the blotting paper or the here in the transfer buffer. So due to capillary action it will get soaked in the transfer buffer. So there are two ways you can do the transfer, either a fully wet technique or a semi dry technique. So in case of wet technique you have to use sponge, and so there will be sponge which is soaked with the buffer, transfer buffer, and in between there will be blotting paper, the membrane and the gel, and again blotting paper and another sponge. In case today for today's demonstration we are going for semi dry transfer. So here we do not need the sponge, we just need the blotting paper which is soaked, and then we will make the sandwich. So I will put some amount of this buffer as well. After the membrane is and the blotting papers are properly soaked, so as I have mentioned that we are going for this electrotransfer. So you may opt out from the electrotransfer, but nowadays everyone go for this electrotransfer because it is quicker. You don't have to wait the whole day or even more than one day. So here what will happen is that we will place our gel and the membrane, and because due to the electricity or the electric field, the protein will come out from the gel and get stuck to the membrane. So this is the instrument which we have. At the bottom part we will take blotting papers. So at the bottom we usually take 4 to 5 blotting papers. Place it here. This should be properly soaked. Then we will put the membrane. Do not touch the membrane with bare hands. We will put the membrane here. Then on the top of the membrane we will put our gel. So in the blotting paper, on the top of the blotting paper I have kept the membrane here. So at the bottom there should be 4 to 5 blotting papers which are soaked in the transfer buffer, and now I will put the gel on the top of the blotting paper. So, as you can see, can see all the lanes and the marker. So, that is why we prefer to put a pre-stent ladder in this case of blotting gel. After you put the gel, then on the top of that again we will put blotting paper. So, this will make a sandwich. Now, in between we do not want any kind of bubble to present between the membrane and the gel. So, for that what we have is a roller. So, if there is any bubble at that particular position, due to this bubble formation, the transfer will be hampered. So, with the roller pin we will just roll out if there is any bubble. There should not be any bubble. If there is a bubble at that particular side, you will see a spot and the transfer will not happen there. Make sure that the gel does not slip out from the middle, otherwise if the membrane is not in contact with the gel, then the protein will not get transferred to the membrane. After you are okay and there is no air bubbles, then we will take the top and we will lock it. So, this is the cassette which we have prepared. In between our sandwich is there for this transfer. Now, here we have this transplot machine. So, basically it will create this electric current and I will switch it on. So, in this machine I can check that voltage and the time till when I have to stop. So, here I will go for this new, and then I will change the voltage to 20 volt and the time to 15 minutes. So, this time and the voltage basically you need to check for depending on your protein. So, I know that in 15 minutes at 20 volt my proteins will all come out from the gel and get to the membrane. But if you see, after you see the gel membrane, if you see that not all the proteins are getting transferred, or if your protein is quite big, then you go for more time. So, now I will put this cassette. So, there are two cassettes which we can put basically. There are two sides, A and B. A I am putting. So, simultaneously you can do two blotting transfer. So, now I have put it in the A and I will go for this run and I will select the A. So, I have made the sandwich and in this blot machine I have started a run for 15 minutes. So, after 15 minutes what we will be able to see is that the gel or the ladder basically, because we can see the protein marker as it is pre-stent, we will be able to see that the marker has gone from the gel will be empty and it has been stuck to the membrane. So, after we see that all the protein bands that are present in the ladder is gone to the membrane, then we will stop. If we take out and see that some of the bands are not properly transferred, then we will again have to transfer it for some more time. So, we will wait for 15 minutes and then we will check if it is transferred to the membrane or not. So, our transfer of the protein from the gel to membrane has been done. And now we will go for the blocking the membrane. So, as I mentioned previously that the blocking is an important step in case of Western blotting, because if there is any part of the membrane which is not blocked, so that will be free where the primary antibody can bind. So, apart from binding to the specific protein against which it was created, the primary antibody will also bind to the free space in the membrane, and that will eventually give rise to this noise that are present after you go for the secondary antibody and the detection of your protein. So, because if your primary antibody is bound to the particular space where there is no protein, but only the free membrane was there, then the secondary antibody will also bind to the primary antibody, because it was raised against the primary antibody, and because the secondary antibody is there, you will get the signal from that particular empty space as well, and it will eventually cause you the error in the judgment, because the protein is not there, but you will still see the band and you will still get the noise. So, now what we will do is we will take out the membrane and check if the transfer is done or not. So, in the gel there is no ladder or no protein is present. So, as I mentioned or shown that it was a pristine marker. So, there in the gel there is nothing. But in the membrane you can see there is a portion where you can see the ladder present. So, basically the protein has been transferred from the membrane gel to the membrane. Now, in a box we will take blocking solution. So, mainly the most commonly used blocking solutions are skim milk or BSA. So, these are non-specific proteins. So, here I have 5% skim milk or fat free dry milk, which was dissolved in TBST buffer. So, this is the buffer later on in every washes, and the secondary antibody, the primary antibody, all will be in the same buffer. So, the milk is also dissolved in the buffer, it is 5%. If you want to use BSA, that will also be to 3 to 5% in the TBST buffer. You can also use the BSA. So, here I will put the skim milk here. And I will put the membrane that has my protein in the blocking solution. Now, I will keep this thing in the rocker, in the rocking condition for almost 2 hours. So, the blocking time is you have to check depending on your protein and the gel, because I am keeping it for 2 hours, because I know after 2 hours it will be enough, the blocking efficiency will be good. But if you see at the later on, if you see there is much more noise in case of your while detecting your protein, you should go for more time in the blocking during the blocking. So, if the blocking is not proper, then there will be noise. So, now I will keep this in the rocker for 2 hours and in the constant rocking motion, and after 2 hours we will wash this membrane and then go for this primary antibody incubation. So, it is 2 hours now and the blocking is done. So, I will remove this skim milk from the membrane. Then I will wash the membrane with 1x TBST buffer, the same buffer which we used to dissolve our skim milk. So, around 3 times we will wash to remove the blocking solution or the protein. You can also keep it for 5 minutes in the rocking motion and wash 2 to 3 times with the solution to sufficiently remove the skim milk. Now, I will put primary antibody. So, the primary antibody is raised against the protein which I want to detect, and this also is dissolved in TBST buffer, the same buffer which we are using here. And the primary antibody So depending on the specificity, the amount which you need to provide in the primary antibody, you have to check. If it is highly specific antibody, then you have to go for 1:10,000 or something like that. So 1:10,000 means in 10 ml you will give just 1 microliter of primary antibody. Or if it is not that specific, then you have to go for this 1:5,000 like that also. So depending on your need, you have to select how much amount or antibody you have to provide, and it is also a trial and error based method. You have to optimize the Western blotting technique. So if you go for one concentration, then you do not see proper bands, then you change some parameters like the blocking, the primary antibody concentration, the secondary antibody, and like that you optimize the whole protocol. So now I will add the primary antibody here. So it is, as I have mentioned, that it is an antibody, so it will specifically bind to the protein of interest, and I will keep in the membrane primary antibody in the rocking motion for 1 hour. Now the time is also variable, because sometimes what happen that the primary antibody is not that specific, or you are not getting that much of signal, then you have to keep it for longer time. So from 1 hour to overnight you can keep it in the primary antibody. So depending on how much time you have, and also what is the concentration and what is the efficiency of your primary antibody, you have to reduce or basically optimize the time in which the membrane will be bind with membrane will be in the primary antibody. Now primary antibody will selectively bind to the protein of interest. If there is a huge number of proteins, there will be different proteins, and they will separate out based on their size. Now when I add primary antibody, the primary antibody will selectively bind to the specific protein, because it has this affinity, or it has this selection for the particular protein. The other proteins it will not bind, and the membrane is also blocked with a non-specific protein with BSA or the skim milk. So the primary antibody will not be able to bind to any of the membrane. So only at the membrane where the specific protein is there, it will get bound to the primary antibody, and then we will wash it and put the secondary antibody. So it's almost 2 hours in primary antibody. So now I will remove the primary antibody. And I will wash it with the TBST buffer. So I will put few ml of TBST buffer, 10 ml almost, and keep it 5 minutes in the rocking motion, and like this I will wash it for thrice. So 3 times I will wash it with the TBST buffer. So this is the last time I am washing. So I have washed the membrane 3 times. Now I will remove the buffer, and next I will put the secondary antibody. Now secondary antibody is also dissolved in the TBST buffer, and since secondary antibodies are very much specific to the primary antibody, you will need only a small amount. So 1:25,000 or so. The secondary antibody, we will keep it for suppose 1 to 2 hours, again in the rocking condition. Now the specificity of the secondary antibody is high, and it will bind to the primary antibody. Also another important feature of the secondary antibody is that it has some conjugated enzyme, so for the detection purpose. So either horseradish peroxidase is there, or alkaline phosphatase is there. So in our case we have HRP, that is the horseradish peroxidase enzyme, and what it does is it reduces, it breaks down the peroxide, and it creates a chemiluminescence. So basically because of the chemical reaction that it undergoes for the substrate, and it breaks down the substrate and produces light, and we can measure the light using blotting as a chemi-doc. Basically the chemi-doc will enable us to see where the light is producing and how much light is producing. So there will be a band at the specific position, and depending on the band we can see, we will detect the position of our protein, the molecular weight of our protein, as well as the intensity, depending on the intensity how much protein is present in different samples. So I have almost 4 samples here, and I want to check how much protein is there in each of the sample. So in the secondary antibody we will keep the the membrane for again for almost 1 to 2 hours, and then we will wash the secondary antibody away from the membrane, and we will put the substrate for the enzyme. So, in our case the substrate basically is luminol and peroxide, hydrogen peroxide. So, those two we have already purchased, and those two substrate we will mix in equimolar concentration, and almost around 100 to 500 microliter we will put on the top of the membrane and wait for 5 minutes. Then we will just take the membrane and put it in the chemidoc machine, and we will see what is the band, and if you just plot it in the chemidoc machine, we will able to see if there is any band is present or not. Now, the membrane is almost 1 hour in the secondary antibody. So, now we will remove the secondary antibody. And we will wash the membrane again 3 times with the TBST buffer. We can keep the membrane in TBST buffer itself. So, we will keep in the PBST buffer 5 minutes at each interval, and after that we will throw away, and then at the end of it we will just take out the membrane and put the substrate for this horseradish peroxidase enzyme, and then we will wait for this development of the band. So, now after the washing is done, we will take out the membrane and put the substrate inside on the top of the membrane, wait for 5 minutes so the enzymatic activity can occur, and then we will take out the blot and start for the development of the blot. Now, there are few things during this Western blotting technique which we should be able to remember. That first you have to run the gel properly, so that all the proteins are separated based on their size. You can either run a native page or a denatured gel. If you are running native page, you will not able to get the molecular weight of the protein, but if you are running a denatured or denaturing gel SDS page, then you will get the molecular weight as well. And after you run the gel, then you cut a small piece where your protein is there, you cut the gel and you place it on the top of the membrane. Now, there are two types of membrane, nitrocellulose membrane and PVDF membrane. If you are using a very big proteins, then you should go for nitrocellulose membrane. If your protein is small or medium size, you should go for the PVDF membrane. Now, PVDF membrane are important and useful in different cases because it is sturdy, it does not break easily. You can reuse it the membrane for long time. Basically, after primary antibody, you can strip the primary antibody and again reprobe the membrane with a different primary antibody. So, if you are detecting in the same gel, if you are detecting two different type of protein, you can do that with the help of this PVDF membrane. While if you are using nitrocellulose membrane, there will be no reprobing and you just can use it once. Now, the membrane is important as well as the how much time you have to go for this electrotransfer, that will depend on your protein. If it is too big protein, then it will take longer time to transfer. If it is a small protein, it will easily transfer. Depending on your protein size and the behavior of the membrane, you have to decide how long you have to keep. So, electrotransfer is a quick way, so it is a useful technique. So, the transblot helps and in 10 to 15 minutes the whole protein will get transferred. After you transfer, you should be able to do the blocking. Now, after you develop the gel membrane basically and see there is a too much of noise is present, then you need to standardize the blocking parameters. The blocking should be done in the BSA or the skim milk solution, and the timing also will differ. So, if you feel that the blocking is not proper, keep the blocking solution for longer time, so that the blocking is proper, then only the background noise will be less. If you are using PVDF membrane, always you should go for proper blocking, otherwise the background noise will be high. If you are using nitrocellulose membrane, then the background noise is not that much, so that in that way nitrocellulose membrane is better. But if your blocking is proper, there should not be any proper any background noise, and also to remove the background noise, the washing steps should be properly done. So you should be wash the blocking reagent away properly, the primary antibody away properly. If there is the washing steps are small or it is not properly done, then again there will be background noise. So do the washing steps properly. If you feel the background noise are much more, change the blocking parameters, change the washing steps, make it a longer time. Now after you do the primary antibody incubation, then you should go for the secondary antibody incubation. Usually secondary antibody incubation takes much lesser time than the primary antibody incubation, because secondary antibody incubation is very much specific to the primary antibody. Now after the secondary antibody incubation, you can go for development, and you can see where the gel or the blot is, depending on the size. There might be some problem with the blotting. So if you see that there is a huge amount of noise, again check the washing steps and the blocking solution. If you see that the intensity of the band is very low, then probably you should keep the primary antibody incubation for longer time, because that might be not very specific to the protein. So keep it for a longer time. Sometimes you will see there is a huge amount of like huge blob of black spots, then there might be few reasons for that you have loaded too much of protein. You should not load too much of protein in the gel. You should check the amount of protein that you can load, and based on that you choose the sample and load that much of protein. If you are loading too much of protein, then there will be oversaturation and the gel will be very bad. Also, if you are putting too much of substrate for this HRP enzyme, then also that will create problem. There will be spots, black spots in the membrane, and you should also avoid that. So a very less amount of substrate is enough to make the gel proper. So during Western blot, you should standardize these parameters. You should go for this different time and different interval, and based on what you feel is proper for your protein, you should standardize the parameter and go for this technique. Now it is a very useful technique, and it is qualitative as well as semi quantitative. If you have different sample, you can get the relative amount of the protein that is present in the sample. If you have an unknown sample and you do not know if a particular protein is present or not, you can detect that particular protein based on this Western blot technique, and it will also give the molecular weight as well as the amount in a particular tissue sample. So Western blot technique is used in the molecular biology lab and mostly in case of protein lab in day-to-day basis, because it is a very good technique to and it is very sensitive as well. So if you want to detect any particular protein, you can detect that.